Hello, my name is Rachit Chandrakul. I'm from Winston-Salem, North Carolina in the US, and today I'm going to be talking to you about my project, establishing an in vitro liver cytotoxicity model for per and polyfluoroalkyl substances, or PFAS, and investigating potential countermeasures. So, as we all know, the liver is a very important organ for performing many crucial functions such as metabolism and detoxification. However, around 80 to 100 million adults in the US have fatty liver disease and are at risk for liver failure and cancer. In just the last year, around 52,000 people died from liver disease and cirrhosis. And out of the various contributing factors, such as diet, alcohol, and smoking, environmental toxins such as PFAS significantly contributed to liver disease and cancer. So, what exactly are PFAS? Well, PFAS are a class of non-biodegradable synthetic chemicals, known as forever chemicals, that are used in many consumer products, from nonstick cookware and waterproof clothing to firefighting foams and even fast food containers. So, they're present in our food, our air, and our water, and out of their various detrimental effects on the human body, they can cause kidney disorders, higher cholesterol levels, immunotoxicity, and changes in liver enzyme functions. PFOA and PFOS are two of the most dangerous types of PFAS due to their longer 8 carbon fluorine chain chemical structures, as you can see on this diagram on the right. So, in this project, I wanted to establish an in vitro model to test PFOS cytotoxicity, because PFOS is the PFAS chemical that I chose to use, using human liver cells, and to establish this model for future drug screening of hepatoprotectic drugs, as well as to determine the protective effect against PFOS using curcumin, which is a natural polyphenol compound. So this is my research study design where I essentially cultured hepatocytes and treated them with PFAS in this 96 well plate arrangement, as you can see on the left, and to in a dose-dependent manner to observe the toxic effects of PFAS. And these were analyzed using cell viability assays like ATP and cytoxgreen. And I also conducted Nile red staining and immunocytochemical staining with these various biomarkers. To test the protective effect of curcumin on the cells, I co-treated the cells with curcumin and PFAS and observed the effects using these similar methods. So as you can see in this graph, the effect of PFAS on hepatocyte viability was tested after 72 hours of treatment of PFAS using the ATP assay, and PFAS was shown to induce cell death in a dose-dependent manner, with the highest doses here at 100 and 150 micromolars, conducing in the greatest amount of cell death. This cytotoxicity is also evident by this host nuclei staining, as you can see in this image on the right with these apparent apoptotic cells. So now I wanted to look at some of the biomarkers of liver injury, and one of the most common biomarkers is excess lipid accumulation and ballooning of the liver cells. So liver toxicity can disrupt lipid metabolism, which leads to this accumulation of lipids in liver cells. And as you can see in this image here, where compared to control, where the green is a Nile red stain for lipid accumulation, the PFAS treated cells show a much higher concentration of lipid accumulation. And the cells are also swollen in size due to this ballooning effect. And this eventually can lead to cellular death. So I also looked at a very specific liver biomarker called HNF4 alpha, which is a target of chemically induced liver injury and is very important for the development and maintenance of liver function. And the loss of HNF4 alpha expression is also associated with the progression of chronic liver disease. So as you can see in this figure right here, compared to control, where HNF4 alpha is expressed in green in the nuclei, PFAS treated cells show a really significant downregulation of this HNF4 alpha expression. And when quantified, that accumulates to around a 50% decrease in HNF4 alpha expression. And this suggests liver injury and stress. Further, PFAS also induces the aberrant remodeling of the actin cytoskeleton. Um, in these PFAS-treated hepatocytes, and this can lead to the promotion of stress fibers. So as you can see in this figure, where the phalloidin was used for stress fibers, here indicated in red, the PFAS-treated cells have a greater concentration or density of these stress fibers, and this is widely recognized as a indicator of oxidative stress. So now that all this data showed how um, the PFAS can induce cytotoxicity in hepatocytes that leads to oxidative stress, I wanted to investigate an antioxidant of the natural polyphenol compound curcumin to counteract this PFAS-induced liver injury because curcumin is widely used in foods and is recognized as having many antioxidant and anti-inflammatory properties. Previous studies have also indicated that curcumin can mitigate liver damage induced by heavy metals, pesticides, and even alcohol. So when co-treated with curcumin and PFAS, as you can see in this graph here, whereas PFAS cause a significant decrease in cell viability, the addition of curcumin helps to maintain cells. And curcumin in itself is not at all toxic to these cells. Further, the co-treatment of curcumin also helps to prevent the accumulation of lipids, as you can see in this figure, where the Nile red signal is significantly less in PFAS and curcumin cells. So this again suggests the protective effect of curcumin. Now, going back to HNF4 alpha, whereas PFAS significantly reduce the HNF4 alpha expression, curcumin helps to keep that upregulated. As you can see in this figure and also in this graph here, the expression of HNF4 alpha is maintained with co-treatment of curcumin, again suggesting curcumin's protective effect against PFAS-induced hepatotoxicity. Now, to understand how this works, how does curcumin exactly protect the cells, is to look at the mechanism of curcumin protection, which is where curcumin basically is known to activate certain antioxidant pathways, such as the NRF2 HMOX1 pathway within cells. So I looked at a very specific biomarker of the antioxidant protein HMOX1, and as you can see here, compared to control, where HMOX1 is shown in green, the curcumin-treated cells have a much higher signal of HMOX1. And this shows, therefore, that curcumin prevents PFAS-induced hepatotoxicity by the activation of this NRF2 HMOX1 antioxidant pathway. So in summary, I was successfully able to establish an in vitro PFAS hepatotoxicity model, which can be used as a tool for drug discovery. And the PFAS cytotoxicity was shown in a dose-dependent manner, demonstrated using cell viability assays and certain biomarkers, showing lipid accumulation and oxidative stress and downregulation of HNF4 alpha. Further, curcumin was tested as an antioxidant, natural plant-derived polyphenolic compound to counteract these toxic effects, and it showed to successfully prevent PFAS-induced toxicity and upregulation of HNF4 alpha and inhibiting oxidative stress. And further investigation suggested that curcumin does all of this by activation of the NRF2 HMOX1 antioxidant pathway. So this model, therefore, can be successfully used for potential therapeutic drug screening for PFAS-induced toxicity. Now, in the future, to further investigate the counter effects of curcumin against PFAS lipotoxicity, certain measurements such as a Ross assay and cytokine assays can be completed. And in addition to curcumin, other natural antioxidant polyphenols such as resveratrol, quercetin, and EGCG can be evaluated using this established cytotoxicity model. Dose-dependent effects of all compounds will be able to be tested, and synergetic effects will also be tested. And after narrowing down potential polyphenolic compounds, this work will be further extended to use 3D human liver organoids to mimic the real in vivo structure and function of a human liver for preclinical in vivo testing session and the stage which can be further applied for clinical applications in the future. So I would just like to thank my mentor, Dr. Gauri Kulkarni, for her support and guidance on this project. the W Firm core members, the director of W Firm, Dr. Anthony Atala, as well as the high school summer research exposure program team, Director Joan Shang, Dr. Carrie Adams, and Brandon Rains for organizing this um, research opportunity, which was funded by the National Science Foundation and conducted at the Wake Forest Institute for Regenerative Medicine. Thank you so much.